Foraging. Foraging encompasses searching for and exploiting food resources as a means of maintaining or increasing an animal's fitness because it plays an important role in an animal's ability to survive and reproduce. The foraging behavior of animals is also a response to the environment where the animal lives, often described in terms of optimizing the highest ratio of energetic gain or payoff versus the cost while foraging. Ants forage in a manner that utilizes the principle of large payoff and low cost, otherwise known as polydomy, by including multiple nests and increasing the colony's foraging success when food sources were randomly scattered in the environment. According to a study performed by Joseph Jun, John Pepper, and Van Savage, labeled Allometric Scaling of Ant Foraging Networks, the function of foraging in ants in regards to the total area foraged by the colony and consequently the resource flow to the nest and the rate of colony metabolism increase non-linearly with the number of foragers. Since the cost of foraging increases linearly, their model predicted an optimal number of foragers and therefore total foraging area that maximizes colony fitness or energy allocation to reproduction. Multiple nests increase the colony's foraging success when food sources are scattered randomly in the environment, not solely confined to a single nest, but inhabiting several dispersed nests, a colony organization referred to as polydomy. This phenomenal behavior is a backbone of ant colonies across the world. Chemical signaling is the main causation that drives ants to exhibit foraging behavior. Unlike social vertebrates, which have large brains and cerebral cortices, ant brains are not bigger than those of solitary insects, but they are more specialized. The biological success of ants is probably not so much the result of an individual's brain as it is the concentrated action of a colony of hundreds or thousands of brains and their response to chemical signals known as pheromones. For foraging ants, it is essential not only to locate food sources, but also to find their way back home. Trails may be marked by pheromones, but many ants rely on visual navigation and probably olfactory and magnetic cues. Besides the perception of stimuli, this requires learning and memory abilities that allow ants to remember odors, directions, and make use of landmarks. According to Ophelia Gronenberg's study labeled Structure and Function of Ants, Strength in Numbers, each olfactory receptor neuron in the antenna, which normally ranges between 50,000 and 60,000 in Formica pratensis, sends an axon, which is a long nerve fiber, through the antennal nerve into the antennal lobe of the brain, where the axon terminates on and interacts with the secondary neurons, or interneurons. Antennal lobes are particularly large in ants for two reasons. Firstly, ants strongly rely on olfaction, more so than any other insects with similar behavior. And secondly, pheromone communication is most advanced and elaborate in ants compared to any other social insect. The antennal lobes process both ordinary odors and pheromones. Olfactory axons terminate in the antennal lobe in the globular structures called glomeruli. Each glomerulus receives input from only one particular kind of sensory neuron, but many individual neurons of that kind, which in turn express one specific kind of odor receptor molecule at the receptive site in the antennal cingulum. Therefore, one antennal lobe glomerulus might correspond to one particular kind of odorant receptor molecule and recognize a subset of odor molecules out of almost an infinite number of chemical compounds. The arrangement between sensory neurons and antennal lobe glomeruli suggests that, in general, the number of glomeruli in an insect correlates with the number of odors that can be discriminated with precision. In principle, the perception and processing of pheromone information does not differ much from the other odorants. Many insects have specialized glomeruli that process different components of sex pheromones, which usually indicates the number of specialized glomeruli that reflects the number of chemical compounds that constitute the pheromone blend. This has recently also been shown for carpenter ants, which suggests that different castes have different thresholds for particularly different odors, which probably results from the differences in the number of receptor neurons for that particular odorant. Ants are social insects of the family of Formidae, which belongs to the order Hymenoptera. According to Edward O. Wilson and Bert Hobdobler in their study The Rise of Ants, a phylogenetic and ecological explanation, ants evolved from wasp-like ancestors during the mid-Cretaceous period and differentiated after the rise of flowering plants about 130 million years ago. Today, more than 12,500 out of an estimated total of 22,000 species have been classified. So there's still more to learn about ants. Hooray!